everyone, this is Nikki Gratrix and welcome to another episode of the Abundant Energy Summit. Today I have another fantastic, experienced and highly qualified practitioner who will specifically speak about mitochondrial function and its role in fatigue. It's Gilian Crowther. She has some really cutting-edge, fascinating information to share, so I'm very excited by today's interview. Gilian is a naturopath and a registered nutritionist who studied complementary therapy in Germany for many years before completing her training in the UK. She encountered cell symbiosis therapy, which we call CST while there, which is a unique and highly effective approach that traces the origins of chronic disease to mitochondrial dysfunction. CST originated over a 30-year research period by the German Dr. Heinrich Kramer and now has a fast increasing network of over 4,000 practitioners spread across Germany, the UK and Europe. Gillian trained as a CST therapist, now works with the Academy of Nutritional Medicine, which is the large network of practitioners with a clinic in London. And she also holds seminars for the CST Academy and translates literature on the subject, including books. So a very warm welcome to you, Gillian, and thank you very much for sharing your time with us today.
Thank you very much, Nikki, for that very kind introduction.
Fantastic. So mitochondrial function and these little organelles called the mitochondria are a huge theme on this summit when it comes, especially as it comes to chronic fatigue. So can you remind us about what these little organelles in our cells are, what they do, and also what evolutionary biology shows where they originated from?
Yes, well, first of all, they originated from bacteria. They appear to have originally been what were called proteobacteria, and round about 2.1 billion years ago, when the first ice age took place. Before then, the entire Earth's atmosphere contained no oxygen at all. It was anaerobic, and the archaea, which were the organisms that dominated at that time, were converting carbon dioxide into methane. And after the first ice age, the oxygen levels began to rise exponentially, and the reason appears to have been that cyanobacteria, that's another kind of bacteria, had learnt to produce oxygen via photosynthesis. And so the mitochondria originated from bacteria that were aerobic. In other words, these bacteria that were producing oxygen via photosynthesis. So they kind of joined up with the archaea, this other organism, and that's where actually the word symbiosis comes from in cell symbiosis therapy. They formed a symbiosis of these two organisms, with one being the host and the other one being a kind of symbiont, in other words, an occupant inside. And in fact, there are very, very many of them. Our cells contain an average of about 1,500, so they're tiny little things. There's about 1 billion you could fit into a grain of sand. So they produce our high-performance energy, and without them, we only have very, very limited energy available. So I'll talk a little bit more perhaps about that switching mechanism, the high-performance versus low-performance energy, in a moment.
Yes. Great. And it's amazing to consider that our mitochondria were in fact coming from this sort of ancient breathing, respiring bacteria that were engulfed by these larger anaerobic bacteria to become basically our cells as we know them, as you mentioned, living symbiotically. What was the advantage to each of them to live in this sort of symbiotic way? How did they help each other sort of with overall survival?
Well, it appears logical, and also phylogenetic studies have sort of looked into this, going back in evolutionary biology. It would appear logical, certainly, that the archaea were threatened in their survival as the Earth's atmosphere began to change and oxygen levels rose, because they were and are primarily anaerobic. That means they couldn't breathe or metabolize oxygen. So it was convenient for them, we imagine anyway, to have a sort of an oxygen respiring organism within them. And for the proteobacteria, the predecessors of our mitochondria, it will have been very practical to have an anaerobic organism to take over when oxygen levels were low. And actually, interestingly enough, we do still have and need these two forms of respiration even now, because there are various situations where we do need to rely on anaerobic respiration. So, for example, when we use more energy than we have available from our anaerobic, from our ATP, that is our adenosine triphosphate, which is our energy currency, which is largely produced, or should be produced largely, by our mitochondria. If we don't have enough of that, then our anaerobic energy generation kicks in, and we can produce a lot more of this anaerobic energy. So that's a good thing when you're running a marathon or whatever. You need that, though it does have as a side effect lactic acid, and that is that burning feeling that you get in your muscles. So that's not meant to, physiologically, it's not meant to continue long term. And then when we have a lot of cell repair after, for example, inflammation, when cells are damaged and need to be repaired, the building blocks of cell repair are made where in the cytosol, that's the part of the cell where you don't have mitochondria, that's where actually these building blocks, nucleotides and lipids and so on, need to be built for that. So it's very important to have anaerobic energy generation for that. And also, it appears that when the cell divides and the gene envelope opens and all the genes are exposed, at that sort of tiny moment, the mitochondrial respiration, in other words, aerobic respiration, downregulates and sort of shuts off, because there's a lot of free radicals which are very dangerous, very harmful, shooting around as you metabolize oxygen, and they would damage the genes. So at that moment, it's a good idea not to have that going on. So there's a kind of very symbiotic relationship going on between these two forms of energy generation. Even now, we need them, but the main form of energy generation should be mitochondrial.
Very interesting. So if I've understood you, the mitochondria produce aerobic energy, and they actually produce higher levels of ATP. But when the cell needs to do things like cell repair or replication, like you said with the DNA replication, we actually want to down-regulate mitochondrial function so that the cytosolic energy production, which is anaerobic, kicks in. But maybe you'd like to share, just for the sake of the audience, with this cytosolic sort of source of anaerobic energy versus mitochondrial energy, would you just like to expand on sort of, they're quite different efficiency levels of energy, aren't they? So I think cytosolic produces much kind of less energy with, as you mentioned, these side effects.
Yes. Well, from 1 mole of glucose, you can get 2 moles of adenosine triphosphate, ATP, that's our energy currency. And that's what happens in the process called glycolysis in the cytosol. So that's still inside the cell, but it's not in the mitochondria. And then those 2 moles of, it's sort of like the net result, those 2 moles of glucose then go on to what's called the Krebs cycle, and that itself is within the mitochondria, as well as another type of, it's traditionally conveyed as an engine, really, with a little piston at the end, the electron transport chain. Along there, together with the Krebs cycle, we can produce, and this is all within the mitochondria, 34 moles of ATP from 1 of glucose, and so together that's 36. So much, much more within the mitochondria. And you've got to remember that we have these 1,500 to often many, many more mitochondria in one cell, and also thousands of these electron transport chains in each mitochondrion. And they're what are producing most of the energy. So Nick Lane, who's just written a new book, he's a tremendous writer on the mitochondria, and he's actually just this year produced a book in which he's calculated that it's possible for eukaryotes, and those are cells with organelles in them. Organelles are those sort of little further creatures, like, for example, the mitochondria and Golgi bodies and so on. They can produce 200,000 times more energy on average than prokaryotes, and those are cells without these other organelles in them. So our cells are, on average, producing around about 200,000 times more energy if the mitochondria are working than if they're not, according to the most recent calculations.
Wow, that's very interesting. So we mentioned there that with the cytosolic sort of production of anaerobic, I think the side effect of that, it's not a side effect, but the outcome, the outcomes of it is lactic acid, which obviously is what we get when we get the burn. I guess so athletes get that. But with mitochondrial function, much more efficient. It's like going into fifth gear by the sounds of it. But we have this electron transfer train that produces all these free radicals. And that's its own problem, where we have to make sure we have enough antioxidants.
Exactly.
Right. Okay. Fantastic. So, it'd be interesting when someone is suffering from some kind of chronic infection or inflammation, then how, in this latest research and in this therapy that you work with, how does the mitochondria respond to this in terms of the switch?
Well, there are two aspects to that. First of all, our mitochondria are capable of being protective, so they can downregulate at times where that is for the greater good of the cell, but they can also be damaged and unable to function. That's mitochondrial dysfunction. So, first of all, if you have an infection or a virus, they've actually evolved to sense threats of that kind. And it's the changes in the electron flow, in fact, for normal metabolism that they'll sense in the form of a voltage drop. And they'll decrease their oxygen consumption and allow the concentration to rise in the cytosol. And that acidity helps to stop the viruses from building further blocks, building blocks that they need for their replication. It can help to kill off the pathogens. And so that's all part of the plan, as it were, though it's not helpful in terms of energy. But there are many other reasons why mitochondria might be unable to function well, like, if, as you've just mentioned, if they don't have sufficient antioxidants, or for these free radicals being continuously produced along the chain, or if you don't have sufficient coenzymes and cofactors for that, for the Krebs cycle as well, and also for the electron transport chain. If you have heavy metals or chemicals blocking the membranes of the little mitochondria, which does happen, there are all kinds of reasons why you might get mitochondrial dysfunction. So if that happens, then you get more energy being produced in the cytosol. It's able to upregulate the energy it produces, but it does produce this lactic acid, which can produce, that's part of the pain in fibromyalgia, most definitely. But there are other side effects as well, because if you get stuck in that form of energy production, anaerobic, and you lose the aerobic energy production, then, and I think we'll be getting onto this, it's not just the energy-related side of things that is affected, but also the cell-to-cell communication aspect. In other words, the signaling aspect of the mitochondria can gradually get lost as well, and that's vital for many aspects of our health.
Fascinating. So I was absolutely intrigued when I read about this work and what CST are actually doing, because it's very interesting that, so if we have a kind of chronic inflammatory sort of thing happening in the body, such as a gut dysbiosis, it's sort of sending the cell into a signal of being in constant kind of need for repair, which would mean we would regulate up anaerobic production and downregulate mitochondrial function. And just to connect for the audience, that I think they probably know this by now, we've talked about it on numerous talks, but really our and I mentioned this with Dr. Sarah Myhill, with this mitochondrial function, when it gets downregulated, we are now rationed with our energy, and this is where when mitochondria fail, that's where someone feels like they've pulled the plug on the energy. So, and where we have rationing, so if we go beyond our boundaries of activity, we will then we could have delayed fatigue. So this core aspect of chronic fatigue syndrome, which is post-exertional fatigue, it's incredible. It comes down to this mechanism that you're talking about. It is the core thing. It's one of the most important things we could talk about, which is absolutely fascinating. Now, I'd love for you to expand on that very special sort of mitochondrial function with a twist. This is a new sort of thing which was very interesting. So it's the work specifically of Dr. Kramer to do with the electron transfer chain, and this aspect that the latest research and his maybe hypothesis that it actually produces an electromagnetic field when it emits photons of light, and the implications of that. Would love for you to share that and the additional implications of a problem with mitochondrial function.
Yes, absolutely. Thank you. Well, there's no doubt at all that electrons are being accelerated extremely fast along the electron transport chain. And there are actually articles out there. I mean, he posited this a long, long time ago, but articles are increasingly emerging now to show that data on living cells do indicate the presence of an electromagnetic field. And there is a very special kind of molecular structure about many of the molecules in the electron transport chain. Many of them appear to be able to absorb light. They sort of behave almost as a small antenna and can receive the electromagnetic waves of passing photons. Now, this is very clear in science, but whether it's actually happening within our bodies was sort of doubted for a very long time. It was almost as though quantum mechanics, quantum physics takes place on a macro level, on a micro level, but on a macro level, human bodies, no, impossible. But of course, when you imagine that you've got 1 billion of these tiny little mitochondria in a grain of sand, and then so many thousands of these electron transport chains in each of them, just imagine how tiny the little membranes are in them. In fact, I'll just read a quotation of Nick Lane's about the field along the membrane, because this is all only just coming out now. There weren't sort of electron microscopes able to get down into such detail on our cells, so that people like Otto Warburg also hypothesized something similar, that the structure of ATP being produced by the mitochondria was different to that in the cytosol back in the 30s, but you couldn't prove it. But now, really, the evidence is beginning to come out. This is Nick Lane in this book that's just appeared this year. It might not sound like much, but because it operates over only 5/millionths of a millimeter, the field strength over that tiny distance is enormous, around 30 million volts per meter. That's equivalent to a bolt of lightning. So that's what we've got in our cells as potential energy, and these special molecular structures which sort of have a complex name, sort of pi conjugated alternating double bonds. You can sort of begin to recognize them. Adenine is the first one that Dr. Kramer many, many years ago sort of homed in on and said, look, that really does appear to be capable of absorbing and emitting photons. And it had always been considered to be an appendix by conventional science. It wasn't in this sort of ATP triad. It wasn't considered to be of any importance. But actually it appears that if our ATP loses the adenine, that's probably the heart or the brain of our mitochondria. In any case, these antennae, as it were, if you do accept that, and it's quite a long sort of complex discussion, but if you do accept that they can transport light within the mitochondria themselves, then what you've got is cell-cell communication of a very, very sophisticated kind that will disappear if the mitochondria go down, as it were, if they fail. And many of the molecules in the mitochondria are photoreceptors, and I could sort of give lots and lots of science on this. People are very, very welcome to kind of contact me and ask if they'd like the scientific references for this, but I won't go into all the names, but many of them do have this photoreceptor quality that means that they can emit and absorb light, and that makes them very, very special. And after all, it is an electron transport chain. It's worth remembering that our ultimate energy generators in our body don't have contact with anything but electrons. No macro or micromolecules at all. It's all got to be made into sort of this essence of electricity, really.
Wow. So that's it's fascinating, and a big theme on the summit connects with sort of we're talking now about physics and getting into actually energy medicine, and it's fantastic. So it's a real core theme on this summit again. It's very interesting because this links with some work. I'm just jumping in, sort of putting this in, but Fritz Albert Popp was the German theoretical scientist who did the original research looking at DNA, and found, I'm sure you know this, but
yes, Dr. Kramer and Professor Fritz Albert Popp have actually worked together, and Dr. Kramer has had many of the sort of plant phytochemicals measured at his Institute of Biophysics in Neuss to work out their actual resonance and compare this resonance with the resonance of molecules in the electron transport chain. Isn't that fascinating?
I love that. Yes, it's hugely interesting. And so we're sort of out there because of Fritz Albert Popp and actually Lynn McTaggart, who have sort of helped popularize his work, was this idea that DNA is a receiver and a transducer and an absorber of photons, and a Fritz Albert Popp. But it was completely new to me, to this really cutting edge research, that it now also looks like ATP is a sort of light emitter and absorber as well. And that's fascinating because now that's an information field level.
Absolutely.
So the implications of an informational field, extraordinary in terms of, that's what you mean about the cell-to-cell communication, because it's potentially also instantaneous communication, isn't it? Or speed of light, right?
Yes, completely. And the cell signaling functions of the mitochondria are absolutely extraordinary. I mean, they're responsible for calcium signaling, or at least to a large extent, the release of these reactive oxygen species as communicators, cell-cell communication, T-cell function and neutrophil activation, which is a large part of our immunity, are regulated by the mitochondria, by all accounts. I mean, I've got lots and lots of references relating to that. Of course, not exclusively, but it seems as though they have a real orchestrator function. And, of course, apoptosis of the cell. That cannot happen if the mitochondria don't release something called cytochrome C, which is the second electron carrier. And if you can't... If the cell can't die, if it can't apoptose, then you can't get healthy new cells being, taking its place. And unfortunately, this is what happens, for example, in cancer, that you have these sort of almost immortal cells that just will not die, but they're dysfunctional. And it's very, very important to be able to trigger apoptosis, and that's exactly what the mitochondria do, but cannot do if they're dysfunctional.
There's a few other major factors that are very interesting about what happens when mitochondria fail, and it's very it's probably useful here in terms of if you could share perhaps the impact on the immune system and heme production and some of these other things that I don't think people realise mitochondria and ATP are involved with, because we just think of it as energy, sort of this limited sort of, it's just energy. No, you just mentioned there's a link to cancer and apoptosis and perhaps some of these other results of poor mitochondrial function.
Yes, absolutely. Well, if our mitochondria do go down, then we're sort of losing function along three different dimensions, I would say. Bioenergetics, which we've already talked about, in other words, the high intensity energy metabolism, and then biosynthesis and signaling. So in terms of biosynthesis, we can no longer synthesize heme properly because the first stage of heme synthesis is in the mitochondria. That's alpha-levulinic acid. And so there's an awful lot of cells in the body that require heme to function, not just hemoglobin. And then cholesterol, that's cleaved in the inner mitochondrial membrane. It's called P450 SCC, and if that's not sort of cleaved there because your mitochondria are dysfunctional, then you're not going to get all the downstream steroids being produced in their proper ratios, and that's everything from DHEA and pregnenolone and progesterone to your estrogens and testosterone. I mean, it doesn't mean you won't have any, because it's never that all your mitochondria all across your body are dysfunctional, but you won't have proper sort of ordered production of these hormones. So it can have a hormonal impact as well. And then in terms of signaling, I've mentioned a few things there. I mean, it's even an O2, an oxygen sensor for vasoconstriction, and nitric oxide, for example, is very, very closely related to the mitochondria. And if your nitric oxide levels aren't correct, or if the levels that are being produced have to be shut down because they're damaging the mitochondria, then that will also have a very, very key effect on your endothelial membranes throughout your body. One other aspect to mention is perhaps detoxification. It's fascinating to realize that the P450, the cytochrome P450 enzymes, which you need in your liver in order to do the first phase of detoxification, contain heme. And if you're not producing heme properly because you've got mitochondrial dysfunction, then they won't be able to work either. So there are many, many aspects of detoxification actually that are affected by poor mitochondria, but that's just one of them that really hit me when I realized the connections.
Yes, that is I agree. So this could explain why some people start getting more chemical sensitive perhaps, or more reactive to things. I thought that was fascinating too, the whole heme connection to the CYP450s. So anything that affects heme production. And I didn't realise heme had a mitochondrial link. I thought it was more linked to things like if someone has that metabolic imbalance, things like pyroluria, which can lead to porphyria, which was another issue. But aside from that, there's also a couple of others. Just thought it would be interesting, the impact on Th1 Th2 imbalance, and perhaps also monoamine oxidase and the neurotransmitter link.
Yes, absolutely. Well, to perhaps just mention the Th1 and Th2 link first of all. They're two sort of arms of our immunity, and Th1 is our sort of intracellular defense that's within the cells, whereas Th2, I mean, this is making it very simplistic, but it's sort of our extracellular defense. It's in between cells, more for fighting kind of parasites and larger organisms that can threaten us. And when the mitochondria fail or have to downregulate, then it has a close connection to our Th1, our intracellular immunity, and that tends to be impaired as well, so that Th2 is upregulated. In other words, we have more of this sort of antibody, extracellular armory there, able to fight for us, but not within our cells. So if you've got intracellular pathogens, whether that's for example, Borrelia, the pathogen responsible for Lyme disease, or Chlamydia pneumoniae, or Mycoplasma. There's a whole host of nasties, that you can have within your cells that if the mitochondria are dysfunctional, you will find it a lot harder to fight. So that's another good reason why it's really important to restore mitochondrial health as well as you can and as fast as you can.
There was another aspect that I thought that was also very interesting that is part of sort of CST therapy that I'd only recently come to learn about, which is this this idea that of high calorie intake through the diet, so say a high sugar diet. I learned recently that can actually overwhelm the mitochondria and can lead to sort of free radicals. So in fact, that could a high sugar diet could directly limit mitochondrial function.
That is true, yes. What the mitochondria do if they're being given too much fuel and they realize that they haven't got enough free radicals to quench, they don't have enough antioxidants to quench the free radicals that would be produced, is they will use what's called an uncoupling protein to allow the energy to kind of puff out, to simply escape in between two of the complexes that make up this electron transport chain. So that's not really very physiological because you're taking in a lot of energy and you're having to let it escape as heat. You're not actually making ATP out of it. When I say you're taking in a lot of energy, you're taking in fuel, but the mitochondria in their intelligence are saying, look, it's going to be harmful for us to have to process this right through to the end of the chain, so we're not going to. And that in itself can lead to mitochondrial dysfunction. What happens is that they gradually begin to close down, as it were, and you can see how this in turn would lead to higher levels of glucose in the blood, which of course the blood doesn't like. Insulin levels begin to rise to try and tuck away that glucose and stop it from being harmful. And so you'll get the beginnings of diabetes, that's metabolic syndrome, and this can lead to diabetes, can be a large component of obesity. Imagine you're eating, the food isn't actually going into your cells to where it should for producing energy. So you're sensing this lack and you're eating more and more, being told you're greedy, but actually you're just attempting to get nutrition and energy from your food and you're unable to. So that is what can be happening, exactly as you said.
Wow, so that's it's very interesting. Would you be recommending more of a paleo type diet because of that, or just a low glycemic diet? Or is what's sort of the CST's kind of idea on that side?
Well, I mean, if you've got an unbalanced diet, what CST sort of certainly taught me, and I think does still teach, is that it's a very good idea to first of all check whether there are food intolerances underlying the imbalance and whether there are foods that should simply be eliminated from the diet for a while, because you can replace them with much more nutritious foods that will actually nourish the I won't say patient, but the person who's suffering. So along with all the other therapies that you'll be looking at, and neurotransmitters can of course be a big part of the puzzle here as well, we do use a very sensitive IgG 1 to 4 test in order to see, are there antigens that are causing the problem, and if so, which are the most serious, and then discuss with discuss whether these can be left out of the diet for a while and replaced. And then we do tend to focus more on foods that contain polyphenols, in other words, these signaling molecules that are able to both absorb and emit light. So phytochemicals, a rainbow diet, looking, of course, at what the person's particular needs are and whether they're suffering from, for example, a skin disorder, or is it an intestinal disorder, or what could be underlying the imbalance. And so I wouldn't say that paleo diet is what every practitioner who's also studied CST would advise, but certainly low glycemic is always helpful. And practitioners have their own ways of approaching diet. Many of us are nutritional therapists, but I wouldn't say there's one size fits all. It depends very much on the person's needs.
Fantastic. And that's then very also interesting that a core part of It's interesting how this is a recurring theme that, and also in CST therapy, the food intolerance and food sensitivity testing is absolutely key, because obviously that's going to lead to the gut inflammation, which in turn we now understand the exact mechanism of, okay, that inflammatory thing is going to cause the switch from mitochondrial to systolic, with all those the cascade effect on everything else. So that's fantastic. It really brings everything together. And also it's just very interesting if we just focus on a little bit more about what you said. We had a fascinating conversation with Dr. Deanna Minnick about the rainbow diet, because that's very much her sort of protocol as well. But we're talking about food in terms of, that also has light, and it has light frequencies. So I thought this was fascinating. In terms of supplements, I'm very interested. You mentioned phytochemicals. Would you expand on some of the types and the names of the phytochemicals? And it's quite amazing that we're looking at the light waves that these might be providing, not just the biochemistry, but the physics, which is therefore rebuilding the energy information in the mitochondria as well.
Yes, absolutely. I mean, it's misleading, we find, to put all the focus on antioxidants, because our cells do need radicals for signaling. And the advantage of polyphenols is that they have this dual function of being able to absorb and emit electrons as needed. So many of the different supplements that we have do contain polyphenols in a sort of synergistic mixture. We don't really work with monotherapies, as it were, with sort of high doses of just one particular ingredient, but with combinations. And a lot of these are, for example, I'll just mention the triad that we have, which is sort of the foundation of our micronutrient therapy, if it is difficult at the beginning of therapy, or even for a bit longer, for patients to actually mix their own foods in their diet to get exactly what we've certainly felt that they probably need in order to to restore mitochondrial function. So one of these in the triad has many of the cofactors and enzymes that the electron transport chain needs to function well. So that's, for example, nutrients that resynthesize and reactivate glutathione, like, for example, alpha-lipoic acid and lecithin and selenium and a grapeseed extract. They all contribute to glutathione synthesis, and that's our key intracellular antioxidant. Then there are others that help to modulate between Th1 and Th2, those two arms of the immune system I mentioned earlier. And often, but not always, it will be helpful to modulate back towards Th1, but of course what you want is a balance. And there's a particular medicinal mushroom called Agaricus blasi-morel, which has been found to be able to do this, and shiitake and reishi, they're wonderful mushrooms that have these properties. Curcumin, of course, is a fantastic anti-inflammatory. Books have been written about that. I mean, that's really very well established, the anti-inflammatory effects. And I'd like to just mention that anti-inflammatory for me now means pro-mitochondrial, because of sort of looking at these pathways. I think that'll be clearer now that if you can fight inflammation, you can actually restore the mitochondria back to health. So curcumin is one of our most wonderful aids in this, but also bromelain and genistein and oak bark extract, and there are a whole load really, including also trace minerals and amino acids in a specific balance. So that's one of them. And then another one is actually a relatively high dose curcumin extract with about four other polyphenols, as well as some trace minerals. And that has a particularly beneficial effect on restoring photon modulation in the electron transport chain, in that if you go with the CST explanation of what's happening within there, cytochrome C, the second electron carrier, is often what's failed, and that is there shuttling electrons between complexes 3 and 4 in the electron transport chain. And if that's stopped working, Interestingly, it has the same or a very similar frequency to curcumin. That's round about 427 nanometers per second. That's been measured in the lab of Professor Fritz Albert Pops in Germany. And it's, the hypothesis, as I would say, it's not been possible to measure this exactly yet, but the hypothesis is that this bridges the short circuit in the photon switch between these two complexes and can help to normalize information and energy transfer within the mitochondria. So curcumin, a wonderful supplement there. And then the third one is one that contains resveratrol, as well as other polyphenols, that optimizes what's called mitobiogenesis. That's the generation of new mitochondria, new healthy mitochondria. So we have many others as well, but I would say that's the kind of core CST therapy to look at those three and see whether they could be helpful.
So that was absolutely fantastic and hugely interesting, and I've really learned something today. I think that what you said there about the polyphenols, I think in my clinical experience, I think a lot of us are probably coming to the same conclusion, but the whole antioxidant sort of craze that happened, I don't think we've seen the results we should have got, really, or the results you would expect.
Absolutely. So you can overdose, actually, on antioxidants. You really can, yes, because we need these radicals for signaling. I mean, Professor Jeffrey Bernstock at UCL here in London, he's been studying this for around about 50 years, actually, the cell-to-cell signaling properties of ATP and how vital it is that, purine signaling, as it were, how vital it is that we can send the right signals in between cells, and often this is done via the radical oxygen species. So having these sort of little smart chips in our cells that can work out when we need those and when we need the antioxidants seems to be far more sensible than just focusing on the one.
Fantastic, very interesting. So I guess any final sort of aspects you want to share? I think we covered most of the kind of causes of dysfunction. I think the only thing I remember from reading about CST, and don't worry too much, we don't need to cover this too much here, but obviously the detoxification side from toxic exposure, that's all part of your sort of therapeutic aspect as well, isn't it? Because that obviously can gum up the mitochondrial function as well, and you guys do work a lot with that too.
Yes, of course. And that's one aspect, I think, where it is particularly helpful to work with tests, just in order to be able to get a compass on what you should be doing first, what in what sequence. And we do use testing. CST does see itself, or pride itself really, on being verifiable and replicable, and to the extent that the patient can afford it, we do very much like to use tests in order to establish how well the mitochondria are functioning, to use that as a marker and see improvement, hopefully within 3 months and then 6 months and so on, but also to work out what could be blocking function. Yes, exactly. And I mean, it's not just the mitochondria that will be blocked. Often it's the cellular membrane itself. But you can identify heavy metals in various ways in the body, and it's not just those. There are chemicals too. All kinds of things, yeah.
Just lastly then, just for sharing with the audience, which is, if you can, and as it would be interesting, just specifically for mitochondrial function testing, what testing specifically do you do just for that? And if you please do name a lab, if there's a reliable ones that you use.
CST has traditionally used peripheral tests in order to work out mitochondrial function, because it's been so hard to actually go into the mitochondria and be absolutely certain that the ATP that you're fishing out, as it were, is mitochondrial ATP and not cytosolic ATP. So the peripheral tests have been lactate dehydrogenase, LDH, its various isoenzymes. They change in the pattern that they have, depending on whether more energy is being produced in the cytosol or within the mitochondria, and then also M2PK, which is pyruvate kinase isoenzyme M2. that one also changes according to the proliferation of the cells, in other words, the building blocks that it's making in the cytosol. So those two we've, over the years, found very, very helpful, and they really do shift as the patient notices a change in their own kind of clinical picture. But increasingly, we're also working with more direct measures of mitochondrial function, such as PGC1 alpha, which is a marker of this mitobiogenesis that I just mentioned, in other words, the production of new mitochondria, and the relationship between mitochondrial DNA and nuclear DNA, because obviously the nuclear DNA will remain the same, whereas if mitochondrial DNA increases, then you're getting new mitochondria, and mitochondrial ATP itself can now be measured as well. So the various labs we use are all in Germany at the moment, and so it is a little hard sometimes to get samples over from England to Germany, because they have to be very fresh, certainly to measure the mitochondria.
Fantastic. So, Gillian, thank you so much for sharing your time with us today. Where can people find out more about CST therapy, and also find, perhaps a practitioner to work with directly, if you'd like to share your details?
Oh, thank you very much. Well, the CST Academy has an English website called the URL is www.cst-academy.co.uk. And then I work together with the Academy of Nutritional Medicine here in the UK, which is sort of the umbrella for CST here. And that has an easy URL, that's www.aonm.org. And we do have a network of practitioners. We also work with doctors and work with other modalities. So probably the easiest would be to contact either me on my email, Gilian at aonm.org, or the AONM email, which is on their website, and then we'll put you in touch with a practitioner depending on where you live, or otherwise contact Germany. If we don't have anybody, they may well know of someone depending on where you live.
Lovely, fantastic. Gillian, thank you so much again. It's great work and fascinating, and it's been a very interesting discussion. I think a lot of people should have learnt a lot from listening to you and this latest research that you're doing. So thank you. And I will also say goodbye to everybody listening. Hope you enjoyed the interview, and take care, and I'll see everybody again soon.